10:07 on Valentine's Day. The coolest proteins that I have been reading about this week. Ferritin, it's a globular protein. It's what stores iron in our blood. All organisms have it. It's made of two different subunits, a heavy one and a light one. It's got 24 different subunits. It's globular, meaning it's spherical. And what it does, it somehow harvests iron molecules, individual iron molecules, roughly 4,500 of them, and stored in its core in a crystalline form mixed up with, I believe, phosphate and hydroxide maybe. It's just amazing how that protein can pick up an individual atom and then give out atoms based on the body's need for iron. Iron's toxic in the body. Another cool protein is hexokinase. Its structure changes right here. It grabs onto sugar, a sugar molecule, a left-handed sugar, of course all sugars are left-handed, and then it bonds a phosphate group onto the sugar by bringing it in with another motif. I'm not sure which area the protein does that, but that's what makes, I guess, a really important backbone for our DNA. Another protein which, well, I've got the molecules drawn here, the molecules the protein makes. They call this protein it's love B actually. It's like a gingerbread man. It's got 4 centers on the top and 4 on the bottom, so it's legs. The bottom 4 motifs lengthen the chain, and the top motifs add the functional group. So what this protein does, it has 8 functional sites, and it works with love C, another protein, with 1 site. It makes 35 chemical reactions, and what it does, it puts together acetic acid molecules, 9 of them. 1, 2, 3, 4, 5, 6, 7, 8, sorry, 1, 2, 3, 4, 5, 6, 7, 8, 9. And it makes a molecule called dihydromonocholine. Then it adds this functional group afterwards, and that is then logostatin. That protein can do that in just a matter of, I think, minutes or hours. But then logostatin, when you add 3 more molecules, becomes simvastatin, which is a cholesterol drug, but it takes Merck like 2 or 3 months to add those 3 reactions to make simvastatin from logostatin, but this protein can do it in just a matter of moments. So you got to picture this protein as a little gingerbread man that manipulates acetic acid molecules in the 35 reactions sequentially to build that like mechano molecule. It's just absolutely amazing. And then we got to look at the adenovirus. It's just a type of virus. It's got an isosochedron outer shell. It's a polyhedron with 20 faces, all triangular shapes, 30 edges, and 12 vertices. And it's very interesting that this protein gets formed, the outer shell gets formed from identical subunits, and as it turns out, this polyhedron is the most optimum shape that those subunits form. And you got to think how amazing it is that those proteins can assemble into that shape. And it's also cool that the golden ratio is hidden in this, because an isosochedron is formed by taking 3 golden rectangles, crossing them at the middle, and then connecting the vertices with new lines. So, 30 planes have 12 vertices, and you connect them with lines, and that's the isosochedron. So hidden in adenovirus, you can extrapolate the golden ratio. The golden ratio is simply that point in the line where A to C is to A to B, as A to B is to A to C, and that roughly brings it right here at 1.6. It's the constant phi. So there's lots of maths and biology, and what I'm struggling to get my head around is how a protein can sense the world and respond to it, recognize molecules. It has to be, I guess, computation run across the chemical bonds and the atoms and the delocalized electrons. Because there's a good reason why the amino acids all have pyrimidine and pyrimidine rings, because they have a lot of delocalized electrons. So there's got to be computation going on in a protein. You got to view a protein as a machine with a function that can run algorithms. That's my take on biology.